Boa noite a todos, meu nome é Bianca, eu sou vice-coordenadora científica do GEAFIP e hoje nós estamos de volta com mais uma palestra. A palestra de hoje será ministrada pelo Dr. Singh. Dr. Singh possui bacharel em agricultura com especialização em proteção de plantas pela Punjab Agricultural University, na Índia. Ele também é mestre em proteção de plantas pela University of Reading, no Reino Unido, e doutor em Plant Medicine pela University of Florida, nos Estados Unidos. Atualmente, Dr. Singh é professor associado e diretor do Plant Diagnostic Center do Departamento de Fitopatologia da Louisiana State University. Hoje, Dr. Singh dará a palestra intitulada The Role of Plant Diagnostic Centers in Safeguarding Agriculture. I want to thank you, Dr. Singh, for accepting my invitation. It's a pleasure to have you here today with us. Welcome to our webinar. Now it is on you.
Hi, my name is Raj Singh, and I would like to thank Bianca for inviting me for this seminar. And I heard there are 13 students in that department, and I think it's going to be a very good seminar. So I'm going to share my screen now. And can you see my screen? Anybody? Bianca? Okay, yes. Okay, so this afternoon, I'm going to talk to you about the role of Plant Diagnostic Center in safeguarding agriculture. Again, I'm Raj Singh. I'm an associate professor here at Louisiana State University, and I'm also the director of the Plant Diagnostic Center. So, as you all know, our population is increasing day by day, and by 2050, it's going to be 9.7 billion people on this planet Earth. That will be a huge number of people to feed. And so to increase that, to feed that many people, we have to increase our production by 70%, which looks like a huge number. So it's very, very important to safeguard the agriculture, safeguard the things that we have from all these invasive insect pests and species, so that we can save some of that food right now that we are producing. As you know, billions of dollars are lost to these invasive pests and pathogens every year. So feeding that much population is going to be a challenge. So it's very important that we start working on saving our agriculture right now. And we have a lot of pathogens that are increasing day by day, and we are seeing more and more pest and pathogen population out there. So for that, we need plant doctors. This is just very recent. I was featured on the Louisiana Agriculture Magazine front page. This is a quarterly newsletter. It's considered a semi refereed journal publication, and you submit an article in it. So if you get a chance to go online on lsuagcenter.com and find this article, you will see some of the diseases that I mentioned about. They're on rise in Louisiana. So why we need that? How do we safeguard it? How many are too many? That's a big question, because every day you deal with something new. There are new invasive plant pests that move from one part of the state to another part of one part of the country to another part of the country, or they move over continents, and then they come to in a new environment where they don't have any natural enemies, and they can cause economic losses and yield and other things. This is an example of last year. This is a commercial grower of tomatoes here in Louisiana, and this is one of our largest growers. And if you can count, there are 12 different diseases that I'm showing you in this picture. And those 12 different diseases, they came from his field during one growing season. Okay, during one growing season, you see 12 diseases. That's a disaster. Okay, and these diseases, once they are out there, they are very difficult to control. So it's very, very important to make sure that we understand these diseases and how to manage them. Sometimes there is complete crop loss. And in this case, this is a watermelon field, about 500 acres of watermelon here in Louisiana, where the farmer or the grower was unable to diagnose the disease because he has never seen it. And when we came in picture, we found out that there's two pathogens rather than one pathogen involved in it. And this was the reason that the management practices that he had before to manage the disease that he had before didn't work, because it was a totally different pathogen that came with the pathogen that was already there. So even his management strategies didn't work because of the pathogen that was new to his fields. And it took us couple of weeks before we can ID that pathogen and let the farmer know. But by the time we were able to release the information, it was already too late, and he lost about 500 acres of his watermelon in just like 2-3 weeks. Another question was, do you have a total crop failure? This is another vegetable grower that we have here in Louisiana, and they are one of our finest vegetable growers out in the state. And even they have issues with some of their crops. As you can see, this is a crop failure for squash. Even they've been growing squash for a long time. When they didn't do all different kind of testing for the field before they can grow it, this is what happens. They had another crop here last year, which had Phytophthora root rot, and that pathogen moved to the squash plants. And as you can see, you don't see any, you see some flowers, but you don't see any fruit. So this is an early crop failure where you have total crop loss, and there is no yield from this type of crop failure. I think you have seen citrus canker before. It is in Brazil too. Sometimes we don't have effective control options. Florida at one point has the largest citrus industry in the whole world. Now they are not, because of this disease, citrus canker. They've been dealing with this disease for about over a century now. It came to Florida in 1910, and they're still struggling with this disease. We had this disease back in 1914, but 1940 it was considered eradicated. After about 100 years, it came back, and we are speculating that this disease, the pathogen, moved with one of those big storms that moved from Florida into Louisiana. And now we have all of the citrus that is produced here in Louisiana has canker on it. All the big parishes or counties that we grow citrus has canker recorded from there. And also sometimes it's difficult to identify. You can see how the two fruit pictures are totally different, but they are same pathogen. So the growers can easily miss if they don't have that kind of information. And on top of that, for citrus canker, we don't have any effective chemicals to prevent the disease from happening. We have some copper bactericides, that you can use in order to prevent it, or like suppress it or prevent it from happening. But our environment is so conducive, we are so hot and humid that you, even if you spray copper, you have to continue spraying it because of the rain events that we have here in Louisiana. And then there is no control option whatsoever. We are very, very unfortunate to have this disease. This is called citrus greening. As you can see, it is vectored by an insect called Asian citrus psyllid. And you can see the different kind of symptom it produces. It's not just one symptom. It produces blotchy mottling, thickening of the veins, lopsided fruit, abortion of the seeds, and twig dieback. And ultimately, the tree will stop producing any fruit, and then you don't have any fruit left on or the tree is useless, and the tree will die. Now, in this case, what happens is that the latent period, or the period from the infection for the tree to produce diagnostic features, it's anywhere from 3 months to 5 years. So if the symptom onset is delayed and you don't see the disease, then there is a latent period, and it can easily spread accidentally to nearby trees because of this insect vector. Again, only control management for this is right now is management of the Asian citrus psyllid. There are scientists who are working on managing, trying to find some other control options that you can use. There are some bactericides like antibiotics, oxytetracycline and streptomycin, that has been extended as a Section 18C for management of the disease. But then some of the institutions like Food and Drug Administration and CDC, they don't approve those use of antibiotics because of the human health. Now, we have a scientist here in California, University of California, Riverside. She developed a peptide from resistant line of Australian finger, and it's just in the news like last week. You should do the Google search and search for citrus greening and peptide, University of California. It will bring you that article, and it's more stable compared to your antibiotics. They are saying that it can be used as a vaccine when in the nursery stock plants. And it also is very easy to spray on the trees. How effective is that going to be? We don't know yet. So once it gets registered for it and comes out in the market in a commercial formulation or in a formulation that we can use, then we'll see how effective this is in managing the citrus greening bacterium. Now, their preliminary results showed that it is very effective. Within a couple of applications of this peptide, they are able to see that the bacteria is suppressing. So that's very promising right now. But again, it's taking time to manage all these insect pests. Sometimes we don't even know what is causing that problem. This was a big greenhouse here in Louisiana, and you can see how this tomato fruit is deformed, and most of his tomatoes look like this. So if you have a crop like this, which is ready to be harvested, and you can't do anything about it, you won't be able to sell this crop anyways, okay? So your entire year's production or entire year's labor and cost is gone, because we can't figure it out what is causing this problem. We had some speculations about this disease, about this problem is because of some of the non-combustible gases that might have caused it. But again, we don't have any evidence. We do have some evidence, but we don't have a proof to prove that this was caused by those non-combustible gases. So there's not much you can do about it, and because it's already there, and you can see that it's at harvesting stage. So there's not much can be done to manage this problem. Distribution and evolution. This graph shows you in 2016 we had, if you look in the inset picture, this is the map of Louisiana, and so the shaded counties or the shaded part of the state is where the 2016 flooding occurred. Okay. So in the graph, it showed you the total samples that we received in the clinic, in the diagnostic center. And the orange is the Phytophthora samples. And then the percentage of samples is in your gray bars. So you can see the comparison before flooding and after flooding. And you can see if out of 924 samples before flooding in 2016, we had 128 samples because of Phytophthora. The number of samples after flooding reduced to 835, but the number of samples infected with Phytophthora went up. And same thing with the flooding, compared to 14% before flooding to 23% after flooding. So you see there is a 9% increase in the number of Phytophthora infected samples. That's a significant percentage increase in the number of those samples. And this was just caused by the flooding. So as you know, Phytophthora moves with flood water, irrigation water. So this proves that those populations may move from one part of the state to the other part of the state within a couple of weeks. So like Bianca said, we have this Plant Diagnostic Center. We are an entity within LSU Ag Center. We call it Plant Diagnostic Center. And we have a webpage after this seminar. If you get a time, you can go and find more information on what we are doing here. We have fact sheets that we produce for our growers, clients. We have management guides. We have other information that are very useful for the growers. Then we have the information on how to submit the samples, how to obtain a sample, and the diagnostic fee and all that. So the website is, if you look on the bottom right-hand side corner, it's lsuagcenter.com/plant diagnostics. So I'm going to show you some of the data from our last year sample numbers. So this is from 2019. Right now we are in a COVID-19 situation. We were closed for about 3 months. We were totally shut down for 3 months. We opened back, and now we are in the phase 2, and things don't look promising anymore. It looks like the number of COVID-19 is increasing again and again. So we might go to phase 1. So I don't have the 2020 data, but this is the 2019 data. And it shows we have about 822 physical samples. That is a large number of samples compared to the size of the state. Then in addition to that 822 samples, we have about 217 digital samples. Digital samples are the samples that are sent in by the agents or some of the commercial growers. What they do is they will take a picture of their symptomatic plants and they send it to me, and I will look at the picture. If I'm able to diagnose a problem from the picture, I will let them know, or I will give them a couple of options in order to that this could be one of the 2 causes, or I will ask them to submit a sample. We do survey with the Department of Agriculture and Forestry here in the state. So the yellow bar shows the number of sudden oak death samples. This is one of the disease that we are really worried about for it to come in the state. This is caused by Phytophthora ramorum. You might have heard of ramorum blight. So this is a sudden oak death, and what happens during this survey is the Department of Agriculture and Forestry, they will inspect nurseries in Louisiana, and they will collect the symptomatic samples, put all those plants on hold, and they will send us the samples. Once we figure it out, if it is not ramorum, then we'll release that information, and then those plants are released for sale. So last year we had 150 of those SOD samples, which is again another big number. Citrus is number 1 crop that we produce here in Louisiana. It's about $10 million citrus industry, but we have some of the major nurseries that grows nursery stock plants, and they not only sell plants here in Louisiana, but also ship plants outside the state to other states. So we had about 123 samples from citrus last year. Like I said, diseases move, and 2018 we had this lethal bronzing that came in, and lethal yellowing came in. So it's also a quarantine disease, high impact disease. Right now we have about, let's say, about $2 to $3 million worth of palms at risk in southern Louisiana. If you've been to New Orleans, you will see how palms are important. They are one of the iconic plant planted in those landscapes. Even with the ongoing situation of the disease, after removing some of the palms, the city is planting new palms to replace those older palms. 130 boxwood samples. Boxwood is another one of the major landscape plant that we have here in Louisiana. All the new constructions, home subdivisions, and new landscapes, new business landscapes. Wherever you go, you will see boxwoods planted in the landscape. And we have this new disease called boxwood dieback, which is spreading at an alarming rate in not only in Louisiana but other parts of United States. And so that also has become a major issue for some of the landscapers and nursery owners here in Louisiana. This slide shows you the the distribution. So the top pie chart shows you type of the commodities that we receive. Our major commodity is ornamentals, 59%, followed by fruits, that 20%. In fruits, we got citrus, we have apple, peaches, blueberries, blackberries, figs, what else? Pecans, okay? And then vegetables, 9%. And in vegetables, we have all the cucurbits that you will grow, peppers, tomatoes. Then you have the winter vegetables. We have eggplants, and the winter vegetable is your cauliflower, kale, broccoli, all those kind of crops. 8% of the trees. This is urban forestry, and we have some major tree issues here in Louisiana. Armillaria is one of the major issue that we see on the mature trees like live oaks and other trees. And unfortunately, there is no control for that. We have this new crop called industrial hemp that we are starting to grow, and we're already seeing major crop loss in that crop. We do get a lot of turf grass samples too, but not as compared to our ornamentals. So the second pie chart shows you the type of diagnostics. Now, you will see the major portion after fungi, you see no detection, 30%, 31%. So these are the samples where the client or the grower will ask for a specific detection, for a specific pathogen to test. So let's say we have a citrus grower and they want the sample to be tested for citrus greening. We will test it for citrus greening, and if it is negative, we will say no detection. So that is why one of the highest number of no detections that we have. It's not like we didn't detect anything out of it. It was for specific detection. And then we have bacteria followed by fungi and no detection. We do see a lot of abiotic issues with the soil profile. In Louisiana, we have clay soils. And if you go about 8 to 10 inches in the soil profile, you will start seeing clay. And you will see after rainwater, it will just stay saturated. So we have a lot of abiotic issues. Our soil pH is not right. The irrigation water is very high in salts and very high in pH. So those things contribute to those abiotic factors. We also do see arthropods, which is insect pest and mites. Not a huge number, but it is there. And obviously viruses, we see some virus viruses too in the diagnostic center. When it comes to our clientele, you see most of our clienteles are commercial as compared to homeowners, and then the agents submit a lot of sample compared to our client. So this is the distribution. And when it comes to the method that we use in the diagnostic center to diagnose that problem, you will see the cultural isolation is at the top with 260 samples. Now, some of these numbers, like some of these samples might have double diagnostic methods. So sometimes what we have is that we'll put everything under the microscope. So microscopy is the number one. Any sample that comes in the lab, I will put that under a microscope. But that number doesn't represent the numbers listed over here. These are the numbers that I used a microscope to diagnose a problem, and I didn't use any other method to do it. But in that cultural isolations, I have used the microscope, but then cultural isolation was my final method to diagnose the problem. So even if you see microscopy at 77, still that doesn't represent the entire microscopy that we do here at the Plant Diagnostic Center. Every sample that comes to the diagnostic center is examined in the under the microscope first, and then followed by different methods. Our molecular diagnostic is also high. You can see that's about 220, very close to the cultural isolation. And this is one of the reasons the number is high, because we do citrus canker, citrus cleaning, lethal yellowing, lethal bronzing, SOD, all, and even our boxwood dieback with PCR. That's our PCR identification, because these are high impact plant diseases, and these are submitted by the agents or the LDAF. So we use that the most reliable confirmation method that we do in there. Okay. So some highlights that we have here in last year. We reported about 5 new diseases in the state. The first one is southern high bush blueberry caused by Sclerotinia sclerotium. It causes the tip dieback, and we have same pathogen causing white mold on parsley. We just published this first report of cotton leaf roll dwarf virus on cotton in the Plant Health Progress Journal. We are working on this Septoria nymphoid peltata. Is an invasive aquatic weed, and it is known to displace the natural native flora and fauna of any water bodies, and it is an invasive weed. So we found that Septoria is one of the pathogen, and that can easily kill it. And then we found a Calthatricum causing anthracnose on Ragula. Last year we reported laurel wilt. That's another invasive disease. Avocado is one of the most important hosts, but here in Louisiana we have sassafras trees, which are mature shade and lumber trees that are under threat from this laurel wilt. It's been reported from 12 parishes last year. Citrus canker reported from two new parishes. East Baton Rouge Parish is where the LSU is, and the next parish, Livingston Parish, is another one that has been tested positive for citrus canker. We reported lethal yellowing from new parishes and lethal bronzing, and also spreading to other parishes from the center of region in New Orleans. One of my students, we are almost finished developing this TACMAN real-time assay for detection of boxwood dieback. I just finished the last version or last editing on this paper, so it will be published in the Plant Disease Journal. I would say we are going to plan to submit it next end of this month, so it should be published very soon. And the reason we developed this method is because in order to identify boxwood dieback with conventional method, it takes you anywhere from a week to 14 days. With this diagnostic method, once you get the sample, you do the DNA extraction, you can have the results within 24 hours. So also this method is able to detect the pathogen at an early stage in the suspected, in the absence of the symptoms. So if the grower has suspected liners that they want to get tested, we can test it with this method, and it's been very, very successful in doing that. It's 100% efficient right now. As a part of my duties, we do the trainings. Last year, I did a 2-day plant diagnostic training for 20 Ag Center Extension agents, where we discussed new and old diseases or emerging diseases, how to diagnose symptomatic from biotic from abiotic disease, disorders. What are the major causes of plant diseases? What are the major arthropods in the state? All that. And we also provide our services to the private sector. I'm sure you are aware of famous Tabasco hot sauce. McElhaney is here in Louisiana, which produces the famous, world famous Tabasco hot sauce. And I've been working with them for last 5 years. And last year we provided them with a field guide to diagnose their problems. We developed an IPM manual for managing insect pests and diseases and disorders of Tabasco hot sauce pepper. And obviously, Bianca was one of my international visiting scholar in plant diagnostic for about 4 months. She spent here in the Plant Diagnostic Center with us. Just to give you an idea, this is the planned number of samples that we received during February of 2020. You can see the variety of things that we receive. You got shallots, date palm, grapefruit, rose, camellia, holly, live oak, tomatoes, some of the ornamentals and Tabasco, ornamental cabbage, dill, mint. So broccoli, you can see it's kind of like very diverse. We don't just focus on one specific crop, and you can also see the cities it is coming from, and then the pathogen and the diagnostics that we do. So you will see, like on these edible fern, broccoli, mint, and dill, we found aphids. So that comes in our other, and then where we see Phytophthora, Phytoplasma, Xanthomonas, again Phytophthora, Armillaria root rot. So this is just a snapshot of February 2020 before we shut down in March, first week of March. We are part of a national plant diagnostic network, and this network was formed in 2002 after the 9/11 event. The reason for this network to be formed was to ensure national agriculture security. And I'll show you what we do with this network. This network is divided into 5 major regional networks. We come under the Southern Plant Diagnostic Network, and our hub lab is in Florida. So all that green, light green area is the Southern Plant Diagnostic Network, and you see the Western, Great Plain, North Central, and Northeastern. And then we have in the center called National Repository Center, which is at University of Purdue. Here is a distribution of all the diagnostic labs that we have here. As you can see, some states have more than one, some states have only one. So in Louisiana, we have only one diagnostic lab in Baton Rouge. Like Texas is a big state, but it has only two labs. When you compare to Florida, they have about five labs, 5 diagnostic labs, because of the diversity of the commodities that they have and the acreage that come under. They have a lot of citrus, they have a lot of winter vegetables, strawberry production, cucurbits, and all that kind of stuff. So when it comes to this, the main goal of this National Plant Diagnostic Center is that once I receive a sample, and let's say I found something new, and we diagnose the problem with evidence that this is true to our knowledge, then we will send that information to our national repository here at Purdue University. When that happens, they have hired staff over there that will look just at the information coming in from all over United States. Now we have Hawaii, and also we have Puerto Rico as part of this network. So once they find out that this is something new, it has never been reported, then they will generate a report. Okay, so this is something that will come next morning to me. So I'm a part of that National Plant Diagnostic Network. So here you see Arkansas reported milkweed bug, and it's there. Okay, so the idea behind that is, if you see any of these labs, okay, any of these labs, they report anything new, anything at all, then that information is sent to all the members of this network. When you do that, you stay ahead of the problem. So let's say I reported something here in Louisiana which is of economic importance. Once we diagnose and identify the problem, we send that information to them, and then once it's confirmed, they will send that information to everybody. So now that information is in Texas, Arkansas, Mississippi, Alabama. So these states are already on a lookout, okay, because that information is already there. So because these are neighboring states, so it is possible that that particular problem that I diagnosed here in Baton Rouge is present in other states too. So before it spreads to alarming level, the states are already checking on their part. They're checking in their state, they're trying to look for that disease, and that's how you stay ahead of the problem. When you stay ahead of the problem, you have time to implement those management strategies. Okay, if you follow the problem, then it is difficult to manage, and by the time you are able to make strategies, by the time you implement them, you are already, the pathogen is already out of the pathogen or pest is already out of the hand. So it's very important to stay ahead of the problem. Now, this network is also something we call it like we are proactive. Okay, we have a network called Be on Lookout, which is that we look for some of the invasive insect pests. So let's say if I receive a sample and it looks very similar to one of the insect that we haven't found in the state, we will definitely make sure that the particular insect that we received in the diagnostic center is not same as one of those invasive pests. So when you do that, there is a lot of non-conformities that you can take off. The data is very, very important when it comes to information like those invasive insect pests. So here you can see another snapshot where all these states have reported something new. So Rhode Island reported hop downy mildew on hops. So all the states around Rhode Island that grow hops, they will start looking for hop downy mildew. So that's how you stay ahead of the problem. This is something, a cumulative number of samples that have been submitted to the National Plant Diagnostic Network since last year. And you can see it's about 1.6 million samples. So this is a very newly developed network. We've been only there for about 18 years, and the first few years was mostly kind of like administration. The actual network started in like 2005. That's when all these samples, they start coming in. So within 15 years, you are seeing 1.6 million samples, and you have about 2 million diagnoses. Like I said before, that there could be more than one problem causing that problem. So that's called double diagnostics. This is the history from, I couldn't get a very recent report. So this is of last year, March. And we started submitting the samples in 2007 to this network when I first started. And you can see within the last 12 years, we submitted about 6,000 confirmed. We submitted a total of 8,813 samples. And again, the detected number is high, but it's those numbers that came in for specific detection. We ran a test on it, and the test turned out to be positive, so we said it's not being detected. So this is a quite large number of samples that we have in the database right now. This is a process that I show to all my audience, diagnostic process. When a sample comes in, the grower is already needing that answer yesterday. So it's already too late for them to submit a sample because they're already seeing the symptoms. So the disease is already out there. So their question is, what is wrong with my plants, or what is wrong with my tomatoes, and how do I correct it? And to answer that question, this is how you go through the process. At the top, you see the detection, diagnosis, and response. Those are the three major events that occur in the diagnostic process. But to make that process happen, you have this another second set of processes that come in. Okay, so FD is your first detector. The first detector is a person who is going to submit us a sample. So it could be an agent, it could be a grower, it could be anyone who is submitting the sample. So they will submit a sample to the diagnostic lab, And each state has a diagnostic lab, like I showed you before. Some have more, others have one or two. So in that diagnostic lab, and this is part of the NPDN, okay? If you just follow me through this process, this part is the USDA and APHIS, okay, United States Department of Agriculture and Forestry, and the Animal and Plant Health Inspection Services. So in the diagnostic lab, we will diagnose that problem, and let's see that problem is something new. Then, in order to confirm that, we send that sample to an expert lab, which is an APHIS lab, and most of our confirmatory labs are in Beltsville, Maryland. Once that information is confirmed, it will go to our state plant regulatory office or state plant health director. This is our Department of Agriculture and Forestry personnel. This is the USDA personnel in Louisiana. After that information, they will generate a response, okay? These two people will generate a response if this is something new. So once that response is generated, it will go to APHIS, okay, which is the Animal and Plant Health Inspection Services, and the State Department of Agriculture. And that the information will also go back to the NPDN, the diagnostic lab, and the first sector. So you can see it is it looks simple, but it is a complex process. There is a lot of different institutions are involved in it. So depending upon how busy those institutions are, or what is the manpower available, and diagnostics can be very easy, or diagnostic can be really late. So it is very complex process. Okay? The reason we have to go through this complex process is we don't want to panic anybody. Okay, because if you release something which is not true, then you lose your confidence. You lose your confidence in the public. So you don't want to do that. You may want to take extra time in order to confirm it, rather than releasing it too immature. What is our mission? Our mission is to safeguard our agriculture here in Louisiana, and every state has their own diagnostic centers. So what we do is we provide diagnostic services to all the plant health problems and their best management practices to commercial and consumer stakeholders. Our commercial and consumer stakeholders, they include vegetable growers, landscapers, nursery owners, fruit tree growers, orchards, golf courses, anybody who can grow a plant. And we achieve that mission by providing them with rapid and accurate diagnosis. And this is very important, that if we misdiagnose a problem, our management strategies are going to be based according to that diagnostics. So if we misdiagnose something, we are going to give them the management strategies which are not going to be effective. Even if they implement those strategies, it's not going to work. Then we do the extension outreach program. Again, this is to teach our growers, teach our extension agents, train our commercial and consumer stakeholders. We do field visits. Last year, I did about 110 field visits, which involve homeowners and also the commercial growers. Again, sometimes you have to go out there because the pictures or the sample is not sufficient enough. Now, when you suspect that it could be a new problem, that's when I like to make those visits. Accurate diagnosis. So I show this picture to almost all my audience when it comes to accurate diagnosis. Now, you see this is a tomato plant. And those symptoms, if you're familiar with the symptomology on these on this tomato plant, these are mostly caused by abiotic disorder like a 2,4-D injury. 2,4-D is a phenoxy herbicide that will cause it's auxin. It's an auxin-related herbicide. It's a growth regulator, so it will regulate the growth of the plant and cause these deformation. So this should happen to the weed, not to the plant, but it's non-selective. So, and it's a broadleaf herbicide. So this plant came in the diagnostic center, and we checked the history, and they said no herbicides were sprayed or anything like that. So we knew from experience, I knew that there could be sometimes pathogens involved in causing those problems. So we tested for tomato spotted wilt virus. At the top, you can see an amino strip, which is a simple form of ELISA, enzyme-linked immunosorbent assay. It's in a form of an amino strip based on the antigen and antibody specificity. So this is specific for tomato spotted wilt virus. So when we tested this sample for tomato spotted wilt virus, you can see how strongly positive this test came out. So these pathogens or these abiotic disorders, they may cause very similar kind of symptoms. So if we hadn't done that, we will attribute it to the herbicide damage. And within herbicide damage, you can't do much about it, and try to be careful when you spray the herbicides next time. But because now this is a pathogen, and as you know, tomato spotted wilt virus is transmitted by thrips. So it's very important if those symptoms are caused by tomato spotted wilt virus. In this case, we confirmed it. Now we have this evidence that these symptoms are, it can be herbicide, but in this case, these symptoms are caused by tomato spotted wilt virus, and we have to manage those thrips so that they don't spread this disease from infected to healthy plants. Sometimes they look very similar. So in this case, you're seeing a pecan tree, a pecan leaf. These are the leaflets, so it has a compound leaf. And you can easily say that this looks like a disease problem. You have yellowing, which is chlorosis, followed by necrosis, which is death of the tissue. And you know this doesn't look like an abiotic issue, because if it is abiotic issue, then it will look very similar. The symptoms on these two leaflets will be mirror image of each other. So in this case, it doesn't look like it is caused by an abiotic disorder. So the symptoms, like we discussed, is chlorosis followed by necrosis, is most probably caused by a plant pathogen, which could be a disease. So in this case, you can easily say it could be a bacterial or a fungal disease, but that's not true. In this case, it is caused by an aphid, okay? And there is one aphid right there. There's one there and one there. The economic threshold for this black aphid is 1 adult aphid per compound leaf. And I showed you 1, 2, 3, and 4. So that's about 400% of the economic threshold. So if the farmer is already behind on how to manage this problem. So insects can also cause symptoms similar to plant diseases. And if you misdiagnose it and you send them the best management practices tailored to a disease, it's not going to be any effective. Rapid and early diagnosis. This is a picture of a late blight of tomatoes. The pathogen is same as late blight of potatoes that caused the Irish famine in 1845 in Ireland. And as you know, that disease caused that famine within like 2 months. The entire potato crop was gone. So we have this pathogen, and this is only 2-day-old lesions. 2-day-old lesions. And you can see how fast it is spreading. So if you miss this window, by the time you are able to diagnose a problem, by the time you receive a sample and diagnose a problem, it's already too late, because the disease already developed and caused the damage. So this is an example of the diagnostic reports that we write. You can see the top is your date and then the client. And then you have the diagnosis. And when I do the diagnosis, I also put a little bit about the pathogen, how it's spreading, what conditions are going to favor that pathogen. Is it a soil-borne pathogen, or those kind of things, okay? So, and then it comes to the management practices. And I always trust or always believe in providing best management practices, including the culture practices, because it's very, very important to have an IPM strategy, integrated pest management strategies. With fungicides or other things, you can manage the problem for a short term period, but when you try to improve conditions that are favoring the disease, then those fungicides are more effective. If you're unable to provide a good environment for your plants to grow, they're going to be stressed out. When they're stressed out, then they're more susceptible to pathogens. So the conditions that are favoring the disease development, it's very, very important to address those conditions before you can start managing that. Once you are able to address those issues, like with Phytophthora, waterlogging condition and poor drainage, if you don't correct those conditions, no matter how hard you try it with other chemical practices, the pathogen is not going to go away. The plant is going to be stressed out, the pathogen is going to stay there, and you're going to waste a lot of time and money in managing that problem. But when you are able to reduce those conditions, when you're able to correct those conditions that are favoring the plant pathogen rather than the plant, if your plants are growing vigorously, if the plants are happy, they're less stressed out and they will be less susceptible to your plant health problems. Extension program in field service. This is a strawberry farm that we visited last year. They had a lot of issues with the plants not growing at the rate they should be growing. So we have one of our agronomist specialists, Dr. Kiki Fontano, standing there, and one of our weed specialists there. So we are trying to figure out why those plants are not growing at the rate they should be growing. And you can see very uniform growth all over the place. So it's very, very important when we go out there, we have enough information so that we can give that information to the grower so that they can start working on our management practices. Then we also do, like this was a field and this is another greenhouse that they have problems with. This is another example that we go there and we'll try to show them. This is kind of like an educational experience for the clients as well as for us specialists too. Training for our extension agents. So this is the citrus greening training that are provided to the agents. And this is an example of just one training. We are planning to do another training on boxwood dieback this April, but the COVID-19 situation prevented us from doing that. But you can see this is like the client, the agents, the extension agents that we have in the field. They have the first-hand experience in looking at those symptoms. So when we train them like that, as you know, a picture is worth 1,000 words. So when they see it in person, and when they go back to their counties or parishes, then if somebody bring in a sample, they can easily diagnose that problem. We do teach master gardeners. I do about 4 of them. These have been started to go online now rather than in person. This is one of the field day, tomato field day. This is one of our largest field day where about 350 people attended. And here I'm showing them the different diseases that are affecting those tomatoes in the field. I do ornamental and turf grass educational programs. We have about 3 major ones in the state. An audience, I would say, about 150 to 175 professionals at each meeting. Then I'll also do the first detector trainings. I just finished one with about 14 advanced master gardener trainers. I do go to horticulture clubs, and if they have any requests, and I do talks there. Workshops, like I showed you before, we just did two-day workshop for the agents at the Plant Diagnostic Center. Now, how do we disseminate this information? We do regular industry updates. Here you can see update on citrus canker, and then you have an update on bacterial leaf scorch of landscape and woody ornamentals. So we do these regularly. Whenever we see that the disease is becoming problem or the disease is spreading to an area where it has never been reported before, we will do those newsletters for the industries to keep them updated on the information about that particular diseases. When it comes to some of the deadly diseases, we do also do the news releases that will go statewide and even out of state with the Ag Center website. And here you can see I did a deadly palm disease detected in Louisiana. That was our lethal yellowing that was detected here a couple of years ago. When it comes to new diseases, it's kind of like when we see, as a plant pathologist or a plant health specialist, when I see something new, I get excited. So this is one of the first report of lethal bronzing detected from Chinese windmill palm here in Baton Rouge. It has never been reported before. I know Florida has a lot of palms, but they never reported this from Florida yet. So this was the first report in the world on palm phytoplasma causing lethal bronzing on Chinese windmill palm. So I was able to publish an Australian plant disease note. So these things are very, very important for your career too. When a new disease come in, we know that this is going to be a problem, then we do the diagnostic guides and alerts. So the boxwood dieback that I was talking to you about. Now, I know it's called when it comes to agriculture, it's not just the food crops, it's everything, because you can't use them for food, but these are for your aesthetic value. And these are also very important for your environment, for your ecosystem. And when you lose these crops, you lose money. So these are also important, not just for the food, but for your agriculture in whole. So we did this diagnostic guide on boxwood dieback, which is very interesting and which is very elaborated. You will find everything about boxwood dieback and similar related diseases in that. At the same time, we do the National Pest Alert. This is one of the IPM source. We will send them the article, and if they think that this is worth a National Pest Alert, then they will publish it and send it to all over US. Sports to the stakeholders. This is one of the things that we provided to the private stakeholders. We just finished a Satsuma cultivar susceptibility to citrus canker research, where we found that our kumquats, especially Miho and Ovari Satsumas, they are highly tolerant to, they are least susceptible to citrus canker. So this is very good information when it comes to the disease problem, the pathogens that you cannot manage properly in absence of the effective management practices. We also found that these two have late onset of the disease. So with other management strategies like copper bactericides and other, like the proper air movement, keeping the foliage dry, and planting these more and more, you have less disease. We also found that these Satsumas have smaller number of lesions of canker and smaller size of the canker lesion, which is directly proportional to your next year inoculum source. So smaller number, less canker lesions, that means less disease next year. As I mentioned before, we just finished the integrated pest management for Tabasco seed production for our Tabasco pepper seed production in Avery Island. They are the world producer of Tabasco sauce. I know most of you have tasted it. In this, we were able to report all the major insect pests, all the major diseases, all the major weed species that they had in the manual, and we took pictures. We wrote the description about that, what kind of damage they will do, everything in that manual. So now they have this manual. They don't have to come back to us. They have this manual. And we also developed a field guide that they can use for their growers or for training purposes to their crew, and they can understand what are the insect pest diseases and what are the beneficials. We do partner with public entities, like with Agriculture Department. So we have a Louisiana Department of Agriculture and Forestry, and we participate with them in sudden oak death, citrus canker, citrus greening, lethal bronzing, lethal yellowing, and boxwood dieback. So they will submit us official samples, and we will provide the identification services to them. And obviously, we provide detection and identification support for high impact pathogens too. So if they don't have a lab in the state, so we provide those services to them. For the Sugarcane League, we provide the seed cane testing services. We just finished the yellow leaf survey program. This year we received about 12,000 midribs that we processed for a tissue blood assay for identifying yellow leaf virus. And obviously we do educational training programs. And one of the training programs that I do is that I teach a class from Southern University that comes in every year. And I tell them the diagnostic methods, the plant pathology, and other aspects of plant health problems. Public partnership continues with NIFA. NIFA is your National Institute of Food and Agriculture. So again, that is our goal, to safeguard national agriculture. Again, this is the NPDN is part of NIFA. Okay, National Plant Diagnostic Center is part of NIFA. Again, that was our major goal. We do the high impact pathogens and pest detections. And this is very important because when those invasive species move into a new territory, they can cause economic losses. And this is one of the issues that when citrus canker moved into Louisiana, the industry was kind of like threatened from it. We had a major hurricane in 2005, which is Katrina, that devastated the citrus industry, and then now citrus greening and citrus canker devastating the citrus industry here in Louisiana. We provide rapid and accurate reporting. Again, this is for our national database system. We are working on the STAR-D accreditation, which is the system for true, accurate, and reliable diagnostics. So under this certification, all the labs at one point will be using standard protocols for diagnosing problems. And I'm an external auditor for this program. I already audit 3 labs. This year I went to Puerto Rico to audit their lab, and last year I audit Iowa State lab. So I'm an external auditor for that. I mentioned BOLO pest alert, which is be on lookout. With that, if there is a new pathogen starting at some place, we are always being alert for that pest, and we stay on top of that to see if there is a problem. Citrus Clean Plant Network program is another program that we work with. It is part of the National Clean Plant Network program. There are several of them. There is blueberries, there is rose, sweet potatoes, and citrus is one of them. Citrus is the largest one that we have right now in that network. And with this network, we have an orchard here in Baton Rouge that we test every year for citrus greening, citrus canker, and citrus tristiza virus. And with that, this is my information. Again, my website is lsuagcenter.com/plant diagnostics, and my email is there. If you have any questions after this presentation or in the future, you are more than welcome to shoot me an email, and I would be happy to answer that question. This is the lab number that you can call. But if you want to reach me, I prefer that you send me an email. That way I'm an extension specialist, so I'm online 24/7. And that's all I have. And let's go back.
Okay.
Bianca.
Thank you for this outstanding amazing lecture. I'm sure everybody could learn from you. Now let's go ahead and get the questions from our public.
Okay.
First question was made by Bernardo. He's asking, how do you work to create new detection methodologies? Are you working any method for greening detection?
So, so the greening detection, so greening is a quarantine disease. Okay. Do you understand quarantine disease? Quarantine disease is when it comes under federal and state regulations. We at LSU are not the regulatory agency. We are an educational entity. So when it comes to a disease like that, citrus greening, for example, or citrus canker, we don't develop methods for them because those methods are developed by USDA labs, And then once those methods are validated, they will send out that information to us. They will share those protocols with us. And then we will implement those methods in our diagnostic center. But we do create new methodologies. Like I just mentioned that one of my graduate students, she just developed a real-time TechMan assay for detecting boxwood dye bags at early stages of the disease development. So we do that, yes.
Now, the next question was made by Christian. The new classification of system to fungi based on phylogenetic species recognition made it difficult to identify fungal species, especially in developing countries like Brazil, due to necessary to sequence many genes in order to identify the species. How are you managing to identify phylogenetic species to diseases caused by many fungal species, like to ones caused by Coleoptera?
That's actually a very good question. So when it comes to diagnosing the problem, remember I mentioned that with that there was a huge number of no detection percentage of samples. So if the client is asking, let's say they're asking for tomato wilt, southern bacterial wilt caused by Ralstonia solanacearum. So we already have that in the state, and we already have that protocol established. So we'll use that method to detect that. Now, we don't do phylogenetic analysis on all the samples that are submitted to the diagnostic center. Now, we will only do, let's say we got a sample from a homeowner and we tested it with an amino strip to test it for Ralstonia solanacearum. And we found out that this is positive for Ralstonia solanacearum. So we'll tell the client that this is Ralstonia solanacearum. Okay. We won't say anything about what is it, race 3 BioWA 2 or anything like that, because we have it's like endemic here in Louisiana. Now, the first reports that I mentioned, so on those we will definitely do the phylogenetic analysis. So the blueberry sclerotium that we found, that was the first in the state. So we are working on a first report on it. And when it comes to first report, obviously you have to do the morphological analysis, and then you have to do the sequencing and the phylogenetic analysis and the cost postulate to publish those reports. So we don't do it on every disease or every sample that comes in because it's not feasible. Okay. Right. Last year we had about a little over 800 samples. So you can imagine we work only over 220 days a year, okay, with all the weekends and everything. So it's not feasible to run a phylogenetic analysis on everything. If we start doing that, we won't have resources to do what we are doing right now. So it's only done on when we see something new. It's never been reported before the state. So we know that we have to work. So last year we had those, and we are right now working on this, and we should be able to submit the aquatic weed paper. It's almost done. So we did the analysis, we did the sequencing on it, we did the bioinformatics on it, and it's ready to be published.
Okay. Emma is asking, can artificial intelligence help improve the efficiency of disease diagnosis?
She has to elaborate that can artificial intelligence. What is meant by artificial intelligence? Can you elaborate on that? What is artificial intelligence? Like, what is she trying to ask? Like, in what aspects? I know that it's not artificial intelligence, but with citrus screening, because it's really hard to diagnose it in the field because of the type of symptoms it produces. They are not unique symptoms produced by a greening. They can mimic other abiotic disorders like nutritional deficiencies. So USDA APHIS program has trained canines, dogs, that they will use in the field. They will go sniff the field, and if they are able to detect those chemicals or the volatiles from that tree, and if they sense it is citrus screening volatile, then they are able to say this is a positive tree. How efficient that is going to be, I do not know, because it's still in the training phase. So depending upon the dog, the age of the dog, how their sniffing power is, what time of the day, how tired the dogs are, it's hard to say. But they're claiming that the dog can detect a citrus greening tree out of a number of trees every time they sniff the tree.
Let's move for the next question. Lucas is asking, what do you think of new technologies to diagnose plant disease? What do you think about high spectral sensors, thermal cameras, and others of large-scale diagnosis?
I do understand these technologies, and I am in favor of these technologies. But at the same time, you have to think that none of these, like if these techniques can be used in the lab, yes, I'm in favor of that. But depending upon the money that we charge to diagnose the problem, give you an example, we charge only $20 a sample. So for large scale diagnosis, yes, we do use like the ELISA. We don't do any other method. For ELISA, for greening, we do those kind of things because like for those 12,000 midribs that we test for sugarcane yellow leaf, we do a tissue blot assay because that's the most economical way of telling if the disease is present or absent. But that will only tell if the disease is present or if the pathogen is present or absent. Now, when it comes to new technologies like your next generation sequencing, those techniques are more appropriate for research labs, not for diagnostic centers, because they're expensive. Okay. Also, we don't have those kind of instruments. Okay. They need training. Okay. So you have to train your staff on it, and they have to go on continuous training to diagnose those problems. And those new next generation techniques, they are not made for all the pathogens. So they're only there for high impact pathogens. So yes, I'm in favor of them. But at the same time, you have to also think about your resources as well.
And Marcela is asking, do you work with nematodes diagnosis, or are there specialized labs for that?
We have a separate lab for nematode diagnostics. It's called Nematode Advisory Services. It's in the same building, but we hired a new person from University of Florida who is our new nematologist. Again, the nematologist is doing it because they are more I have nematode training from University of Florida, but again, the resources, they are in a separate lab. So when we receive a sample, let's say we receive a tough grass sample, and I see that there are root knot nematodes on it, or I see there is sting nematode on it. So I already know that there is a nematode on it, but I also want to know what number of nematodes are present. So at that time, I will send it to the Nematode Advisory Service, and they will count the numbers for me, and they will give me the numbers, and then I will write the report based on those numbers. If the numbers are high enough to cause a damage to the crop, or the numbers are not high enough to cause the damage. So it is done by a separate lab, but we write the final diagnosis on our samples.
Okay. Unfortunately, our time is short, and we are moving for the last question.
Okay.
The disease risk systems works well there? How the farm gets the disease risk alerts? By email, cell phone?
Okay, so like I mentioned, that we have the system where we send out the industry updates. Sometimes I will do an advanced disease alert. We do it through the National Pest Alert, but we also, like for our watermelon growers, we have this CDM IPM pipe website, and every training when I do it with the watermelon growers, I showed them those CDM maps. I showed them that Every time they start their crop, they need to be subscribing those alerts. So we do it that way. We do the news alerts when we see a new disease. We send the news alert to Ag Center, and then they will release that information for the entire state. And from there, the local newspapers, the local radio stations, they will pick up that news and they will publish that in their newsletters too. For our extension specialists, for our extension agents, we have this email web list that I will send that alert to them so that they can also write that in their local newsletter for their parish.
We receive one more question.
Okay.
What to do for diagnose plant virus?
That's a very good question. So we have some of the ELISAs. We get those ELISA kit from Agdia. This is a company. Sometimes we have ELISA kits for specific pathogens like tomato spotted wilt virus, tomato yellow leaf curl virus, cucumber mosaic virus, tomato mosaic virus. So we buy those diagnostic kits from them. Sometimes, like we did the cotton leaf roll dwarf virus last year, so that is a new report. They didn't have an ELISA for that. So in that case, we go to RNA extraction, we do the reverse transcriptase, and then we do the PCR on that. We did the rosette disease like that a few years ago. So we use all different kind of methods. But when it comes to viruses in the lab, if we have the ELISA method available, we will make use of that ELISA method.
Okay, thank you again for being here this afternoon, sharing your knowledge with us. Have a wonderful evening. Good night, everybody.
Thank you for having me. Thanks.
Thanks.